Welcome to the Explainer. So, I want you to imagine your body as this massive, hyper-efficient energy factory. Inside this factory, there are literally billions of chemical reactions going off every single second. You've got these metabolic pathways acting like intricate, high-speed conveyor belts. But what happens when just one microscopic gear breaks down? Just one faulty enzyme. Well, it doesn't just cause a minor hiccup. No way. It can trigger absolute systemic chaos. Today, we're going to unlock the secrets of this energy factory by exploring the metabolic disorders of glycogen and carbohydrates. We're going to hunt down the exact broken gears that cause these conditions and see exactly how they manifest in the human body. Let's get into it.
Okay, let's dive into this. Here is our roadmap for today. We're going to start by looking at glycogen storage diseases. Then, we'll move into some key GSD showdowns, shift gears over to galactose disorders, and finally, wrap the whole thing up with fructose disorders.
All right, Section 1, Glycogen Storage Diseases. Let's check out the glycogen warehouse.
So we can actually functionally classify these broken gears into two main camps, basically based on where the defect hits the hardest, the liver or the muscle. Now, when the liver's machinery is broken, like we see in types I, 3, 6, and 9, the primary issues you're going to see are severe low blood sugar, or hypoglycemia, and a massively enlarged liver, which is hepatomegaly. The liver simply can't release the glucose it has stored up. On the flip side, when the muscle machinery is broken, like in types 5 and 7, well, the symptoms change completely. Instead of low blood sugar, these patients experience profound exercise intolerance, really painful muscle cramps, and sometimes even muscle breakdown. It's a totally different clinical picture.
Moving right along to Section 2, Key GSD Showdowns, where we compare these disorders side by side. Notice the stark contrast in these two pathways right here. We're putting GSD1, which is von Gierke's disease, up against GSD3, known as Corey or Forbes disease. In GSD1, the patient is missing a really critical enzyme called glucose 6-phosphatase. And because of this, both their glycogen breakdown and their ability to make new glucose, that's gluconeogenesis, are just totally defective. Those backed up pathways lead to massive lactic acidosis and high uric acid. But check out GSD3. Their lactate and uric acid levels are completely normal. Why? Because in GSD3, only the glycogen breakdown is broken. Their gluconeogenesis is actually totally intact. This is huge because it means these patients have a built-in lifeline. Giving them a high-protein diet can actually prevent their hypoglycemia.
And honestly, this brilliantly illustrates one of the most fascinating anomalies in all of human metabolism. We're talking about Pompe disease, or GSDI. I find this one incredibly interesting, just because it totally breaks the rules. See, in all the other GSDs, the glycogen simply pools out in the general fluid of the cell, the cytoplasm. But Pompe is uniquely a lysosomal storage disorder. The glycogen gets trapped specifically inside the lysosomes, all because of a deficiency in the acid alpha-glucosidase enzyme. And because this buildup happens inside these little cellular recycling centers, it physically destroys the muscle architecture. In infants, this leads to a massively enlarged heart, hypertrophic cardiomyopathy, and rapid respiratory failure if they don't get specialized enzyme replacement therapy. It's really serious stuff. Okay, let's look at another showdown. This time, we're focusing on the muscle GSDs, McArdle disease, which is GSD5, versus Tarui disease, GSD7. If you've got a patient with McArdle, they lack muscle phosphorylase. The moment they start exercising, they get terrible cramps. But, and here's the crazy part, they experience this amazing second wind phenomenon. If they just briefly rest, their body switches over to burning fatty acids, and boom, they can magically resume exercising. For them, downing a sugary sports drink right before a workout is incredibly helpful. But if you try that exact same sports drink trick on a patient with Tarui disease who lacks the phosphofructokinase enzyme, you have literally just walked him into a terrible trap. They experience an out-of-wind phenomenon. Those carbs actually spike their glucose, completely drop their lipolysis, and deprive their muscles of alternative fatty fuels. You end up making their exercise intolerance much, much worse. Now, let's look at one more crucial comparison that frankly trips a lot of people up. Danon disease versus PRKAG2 syndrome. So both of these conditions can cause an enormously enlarged heart and a very specific irregular electrical pattern on an ECG called Wolff-Parkinson-White, or WPW. But here is your ultimate diagnostic pearl, so remember this. If you see a young male presenting with severe hypertrophic cardiomyopathy and that WPW pattern, it strongly points to Danon disease.
Danon is an X-linked lysosomal storage disorder. That means it severely affects males early in life. PRKAG2, on the other hand, is an autosomal dominant condition and is not a lysosomal storage disorder, which generally means it offers a better survival rate.
All right, Section 3, Galactose Metabolism Disorders. Let's look at milk breakdown issues. Now, what's really interesting about this next concept is this life-or-death clinical clue. Pay close attention to this. E. coli sepsis in a neonate who is feeding equals classic galactosemia until proven otherwise. Literally until proven otherwise. When a newborn starts feeding on milk, they take in massive amounts of lactose, which breaks down into glucose and galactose. If they lack the GALT enzyme, this highly toxic galactose 1-phosphate aggressively builds up in their system. And for reasons we are still trying to fully understand, this metabolic crisis severely impairs white blood cell function. It basically makes these infants incredibly susceptible to gram-negative bacteria, especially E. coli. Spotting this infection early is an absolute massive red flag. But let's talk about the trap of classic galactosemia. I mean, logic dictates that if milk is the poison, taking the milk away completely cures the disease, right? Unfortunately, no. And this is a crucial concept to grasp. Even if you catch it on day one and immediately implement a strict galactose-free diet, long-term complications stubbornly persist. The early metabolic damage, or perhaps the lack of endogenous galactose production, still takes a major toll. These patients commonly develop motor speech disorders, learning disabilities, tremors, low bone density, and strikingly, 80 to 90% of women with this condition end up developing primary ovarian insufficiency. So the diet absolutely saves their life in infancy, for sure, but it's not a perfect cure-all. And just to show you how wildly precise metabolic management has to be, let's look at type 3 galactosemia, or GAL deficiency. In the classic type we just talked about, you completely eliminate galactose. But in GAL deficiency, you must restrict, but absolutely do not eliminate it. I know, it feels totally counterintuitive, right? But here's the underlying mechanism. Patients with GAL deficiency physically cannot synthesize UDP galactose from glucose on their own. And galactose is an absolutely essential building block for structural glycoproteins and glycolipids in the human nervous system. So if you were to completely eliminate it from their diet, you would literally starve their developing brain of structural materials. You have to find that perfect balance.
Okay, moving to our final stop. Section 4, Fructose Metabolism Disorders, the hidden sweet dangers. Just look at the extreme stakes between these two conditions. In hereditary fructose intolerance, which is caused by an aldolase B deficiency, when an infant is introduced to fruit or juice, a highly toxic metabolite called fructose 1-phosphate accumulates. And this stuff acts like a biochemical wrecking ball. It rapidly depletes the cell's ATP, its main energy currency. It forcibly inhibits both the breakdown of glycogen and the making of new glucose. This leads to rapid, severe hypoglycemia, vomiting, lethargy, and eventually liver failure. In fact, older patients who survive childhood with this condition often develop a natural protective aversion to sweet foods. But let's contrast that with essential fructosuria, caused by a fructokinase deficiency. Because the enzymatic block happens way earlier in the pathway, those toxic phosphates never even get the chance to accumulate. It's completely benign. The unmetabolized fructose just passes harmlessly into the urine. It's the exact same sugar, but entirely different outcomes just based on which gear in the factory is broken. So, what does this all mean? Well, it brings us to a fascinating thought to end on. What hidden genetic fingerprints might be dictating how your own body processes its next meal? As you've seen today, tracking these tiny, specific lab abnormalities, whether it's lactate levels, uric acid, or just the presence of a specific sugar in the urine, reveals the extraordinary, super delicate balance of human metabolism. Every single piece of fruit, every glass of milk, every carb you eat, it's all navigated through this massive invisible factory inside your cells. When it works, it's literally a miracle of biochemistry. But when it breaks, it requires masterful clinical detective work to bypass those broken gears. It really makes you appreciate the invisible machinery keeping you running every single day. Thank you so much for joining me on this explainer. I really hope it brought these complex pathways to life for you. And as always, keep learning.